Welcome back, ladies and gentlemen. So today we're going to spend just a little bit of time talking about that idea of homologous chromosomes. And really I wanted to start here by looking at karyotypes, because this is one of the clearest places that we can actually see the display of those chromosomes from a cell. So a karyotype is a way for us to divvy out all of your chromosomes. Typically, we'll do this during metaphase. We'll get a blood sample, run it through a centrifuge, pick up just your white blood cells, um, and be able to fix them, stain them to a slide, and then be able to, as you can see here on the monitor, she's looking at those and basically pulling them out to create an image like this. Now this one happens to be colorized to make the bands even easier to see, and we typically go from the largest chromosome all the way down to your smallest, and then the last chromosome set here is your sex cells. So we pair up those homologous pairs, right? So one of these arms is going to come from your mother, the other is going to come from your father. And these karyotypes are particularly useful in seeing a number of abnormalities, whether it's chromosome number, whether we end up seeing that some chromosomes are too short or too long because of inaccurate replication. Um, Again, too many, not enough. We do karyotypes. They've become more common now that we have better genetic testing, better screening. We can even do karyotypes on we can do karyotypes which are more common in fetal medicine. Um, so it was not uncommon when there was known risk factors to take things like amniocentesis, so taking some amniotic fluid or some parts of the placenta and running karyotypes on babies who we were concerned might have things like Down syndrome, where they've got a 23rd chromosome, they've got an extra chromosome. They should only have two, they've got three. The cool thing is now that we are actually being able to do this even more because we have found that some of baby cells actually cross the placenta and end up in mom's circulating blood. So the ability to do things like the amniocentesis or the CVS, which would take part of the placenta, were at a much higher risk for potential spontaneous abortion. And now that we know that these blood cells or these cells period from baby end up in mom's circulation, um, at least by a certain period of time in the pregnancy, means that we can do these sorts of genetic screens without as much invasive procedure as it used to be. Now it's just a blood draw from mom. They're not always completely accurate. Sometimes they'll think they have baby cells and it's not. So there's potential for additional screening if they see any abnormalities. Um, but it's a cool place to start. So when we look at those karyotypes, again, the banding patterns, the sizes should all be identical from the one that mom gave you versus the one that dad gave you to create these karyotypes. It's pretty cool to see. So when we look at these karyotypes, we look at these chromosomes, we distinguish them into two categories. I mentioned your sex chromosomes, which is your 23rd pair. And in a karyotype, they'll always come down here at the end. So if I'm a male, I'm XY, all right? And if I'm female, I'm XX, which does mean, by the way, when we split out these pairs, dad is the only one who can pass on that Y. So dad is the one who ultimately determines the offspring's sex. Mom can only ever pass on an X. So you got your X chromosome from your mother, and if you're female, your second X came from your father. If you're male, your Y chromosome came from your dad. Um, so those are your sex chromosomes. They're going to ultimately determine the biological sex of the offspring. The other pairs, so in our case as humans, pairs 1 through 23 are your autosomes. And those autosomes contain the vast majority of the rest of your genetic information. Those sex chromosomes, for the most part, are just sexually determining pieces of information. That's not completely true. The X chromosome definitely carries some other genes on there as well. Y chromosome, though, that's definitely the case. It's pretty much male determination genes, and that's about it. So when we get to the S phase in interphase, your DNA synthesis, when this has occurred, we're looking at all of those chromosomes being duplicated, right? And so you'll actually have an N of 3 or a 2N of 6 once we've gotten through that S phase because you have those sister chromatids. And it's going to be important now to really trace and understand where maternal information versus paternal information comes from as we work through meiosis. Where mitosis, it was just kind of understanding, okay, that everything duplicates and then everything splits equally. We're going to see that now it's going to be important to really trace in the production of our gametes where maternal information versus where paternal information goes. So where did the information that your mother gave you end up in your gametes before you pass it on to your children? And same thing for your father's information that he gave you.